All right, let's put on our detective hats. Today, we're cracking a really fascinating medical case where our own red blood cells are the victim. And the mystery? Well, they're not doing their job, but on the surface, they look perfectly normal. Let's start gathering some clues. So, what is going on here? This question gets us right to the heart of a condition called Normocytic anemia. It's a genuine puzzle because when you look at a standard blood test, the size and the color of the red blood cells seem totally fine. And yet, the person is anemic. Something is obviously wrong, and our job is to figure out exactly what. Okay, here's our game plan for this investigation. We're going to start with the initial puzzle, then we'll look at cases where red blood cells are actually being destroyed. From there, we'll identify the suspects, investigate problems with production, and finally, we'll wrap up with the key diagnostic clues that'll crack this case wide open. All right, first things first in any investigation, right? We've got to define the problem. We need to understand what we're actually looking at and identify that first major fork in the road. And the name itself gives us our first big clue. Normocytic just means normal size. Normochromic, that means normal color or normal hemoglobin content. So the cells look totally fine, which makes this whole thing a real head-scratcher. We have to dig a lot deeper. Okay, this is our first big decision point. We examine the evidence, and the number one clue we're looking for is something called the reticulocyte count. Think of retics as baby red blood cells. If that count is high, you know the bone marrow factory is working overtime, pumping them out like crazy. It's desperately trying to replace cells that are being destroyed. That sends us down the destruction path, which is called hemolysis. But if the count is low, that tells us the factory itself might be the problem. So let's go down that first path. Let's assume the red blood cells are being destroyed. This process, as I mentioned, is called hemolysis, and you can think of it as the crime scene of our investigation. But as you're about to see, there are two totally different kinds of crime scenes. And this is where it gets really interesting. You've got two main scenarios. First, there's intravascular hemolysis. This one happens right inside the blood vessels. It's a messy public takedown. Red blood cells just pop. They burst open, spilling their guts everywhere. And that chaos leaves behind some telltale evidence, these shredded little cell fragments called schistocytes. Then, on the other hand, you have extravascular hemolysis. This is way more discreet. It's happening behind closed doors, mostly in the spleen. Here, the body's cleanup crew, these cells called macrophages, are just methodically pulling damaged red blood cells out of circulation. The evidence is different too. You find these perfectly round but unhealthy cells called spherocytes, and often the spleen itself gets bigger from all the extra work. Okay, so we've established that destruction is happening. The big question now is why? Is something wrong with the red blood cell itself, an inside job, an intrinsic problem? Or is some outside force attacking it, an extrinsic cause? Let's dive into the suspect files. First up on our inside job list, hereditary spherocytosis. In this case, the red blood cell's internal skeleton is just faulty. It's got bad bones, so to speak. And because of that, instead of being a nice, flexible disc, it balls up into the small, rigid sphere. Now imagine that little sphere trying to squeeze through the spleen, which is basically the body's super strict quality control inspector. It can't do it. It gets stuck, flagged, and then destroyed. Case closed. Suspect number 2 is G6PD deficiency. This is a genetic thing where the red blood cells are missing a key protective enzyme. They're left defenseless against something called oxidative stress. So when they run into certain triggers, like some drugs, infections, or famously fava beans, the hemoglobin inside gets wrecked. It clumps up into these things called Heinz bodies. Then, as these damaged cells pass through the spleen, macrophages literally take a bite out of them to remove the Heinz body, leaving behind these awesome-looking bite cells. And seriously, how good is this mnemonic for remembering G6PD? Stress makes me eat bites of fava beans with Heinz ketchup. The stress is the trigger. Eat bites for bite cells. Fava beans are a classic trigger, and Heinz for Heinz bodies. It's just perfect. And our last big inside job suspect is sickle cell anemia. It is absolutely wild to think that changing just one single amino acid in the hemoglobin gene causes all this chaos. When oxygen levels are low, this abnormal hemoglobin gets sticky and forms long rods, forcing the cell into that rigid, crescent, or sickle shape. And these sickle cells, they're a total disaster for blood flow. They're stiff, they're sticky, and they get stuck in tiny blood vessels, creating these painful logjams all over the body. This can cause some serious pain and damage to organs. Over time, it can wreck the spleen so badly that the organ just gives up and dies. It's a process called autosplenectomy. Okay, let's switch gears and look at the external attackers, the extrinsic causes. First on the list is autoimmune hemolytic anemia. This is a classic case of friendly fire. The body's own immune system gets confused and starts making antibodies that tag its own red blood cells for destruction. The smoking gun here? A positive Coombs test. That's what finds those antibodies stuck to the cells.
All right, this next one is just brutal. It's pure mechanical destruction. It's called microangiopathic anemia. What's happening is that red blood cells are getting physically shredded as they try to pass through small blood vessels that are all clogged up with tiny clots or other blockages. The result? You get these torn up fragments of red blood cells called schistocytes. They even look like little helmets sometimes. It's a dead giveaway that something is physically tearing these cells apart.
Okay, let's hit pause on the whole destruction angle for a second. Let's rewind. What if that reticulocyte count was low? What if the red blood cells aren't being destroyed? What if the problem is that the factory, the bone marrow, just isn't making enough of them in the first place? This takes our investigation down a whole different but super important path. So when it comes to production problems, we have two main culprits. First, anemia of chronic disease. Here, long-term inflammation makes the liver pump out this hormone called hepcidin. And hepcidin is basically a jailer. It locks up all the body's iron in storage, so it can't be used to make new red blood cells. The crazy thing is, the body has tons of iron, it just can't get to it. Then you have aplastic anemia, which is way, way more serious. In this case, the stem cells in the bone marrow, the master cells that make all our blood cells, they just fail. The whole factory shuts down. And that means you're not just low on red cells, you're low on everything. White blood cells to fight infection, platelets to stop bleeding. This all lines are down situation is called pancytopenia.
So we've checked out the crime scenes, we've looked at the suspect profiles, and we've investigated the factory itself. Now it's time. Let's pull all the evidence together and solve this thing once and for all. So here's the bottom line, the big takeaway. Looking at the cells under a microscope, the blood smear, it tells you a story. It's how you figure out if you're dealing with destruction or a production problem. You see spherocytes? You're thinking about a weak cell structure or an immune attack. You see schistocytes? That just screams mechanical damage. Sickle cells? That's a direct line to a genetic mutation. And if all the cell counts are low, that pancytopenia we talked about, you know the issue isn't out in the body. It's a total crisis back at the bone marrow factory. Each clue you see points you right to the culprit. So you see, by following the clues from that first reticulocyte count all the way to the specific shapes of the cells on a slide, we can untangle this whole complex puzzle of normocytic anemia. The answers are all there. You just have to know what you're looking for. So, what medical mystery should we dive into next?